So yeah, in today's video, we are going to learn about the glyoxylate pathway. So before going into the topic, we should know what is the meaning of glyoxylate. So the name itself says glyoxylate is nothing but salt of glyoxylic acid. So salt of this glyoxylic acid is called as glyoxylate. Glyoxylic acid is also called as oxaloacetic acid, which is written as OAA. In the entire video, it will be shown as OAA. So glyoxylic acid or oxaloacetic acid both are the same. So salt of this glyoxylic acid is called as glyoxylate. And glyoxylate is a major compound where it is broken or synthesized in this entire pathway. Hence the name of the cycle or this pathway is called as glyoxylate pathway. So what does this glyoxylate pathway deal about? We all know that when seeds germinate, they give rise to the shoots root system. So until the first shoot or the first pair of leaves come, the seeds cannot photosynthesize. As they are lacking chlorophyll or the green pigment, the photosynthesis cannot happen. So this glyoxylate pathway is the only solution through which they can obtain energy or they can derive energy. So for their derivation of energy other than photosynthesis, the pathway they follow is glyoxylate pathway where the fats is converted to carbohydrates. So through this process, they obtain the energy for their development, and when the leaves develop, the glyoxylate pathway ceases and the photosynthetic pathway begins. So this glyoxylate pathway deals in the conversion of fats to carbohydrates. So this is the main process where the glyoxylate pathway deals with the conversion of fats to carbohydrates. Since the seed is rich in reserve food material, with such as fats and other things, the fat gets converted into carbohydrates and breaking down of this yields energy for the metabolic activity of the embryo or the seeds. So this glyoxylate pathway is seen in plants, fungi, bacteria and lower invertebrates. It is completely absent in vertebrates or in animals. It is absent in humans also. So it is seen only in lower invertebrates, bacteria, microorganisms, fungi and plants. So this glyoxylate pathway was discovered in the year 1957 by two great eminent scientists, Hans Krebs and Hans Korenberg. These two scientists discovered this glyoxylate pathway. Hans Korenberg was the student of Hans Krebs. Krebs, we all know, was a famous discoverer of the Krebs cycle. So Hans Krebs and Hans Korenberg discovered this pathway called as glyoxylate pathway, which involves a conversion of fats into carbohydrates, especially in plants and fungi. So it was first discovered or reported in Pseudomonas species. It is a bacterium, we all know. So it was first reported or discovered in Pseudomonas species using radioactive labeled carbon 14. So using radioactive carbon 14, they discovered this pathway in Pseudomonas species. So where does this occur in case of plant? In plant, it occurs mainly in glyoxysomes, mitochondria, cytoplasm or the cytosol. What are these glyoxysomes? So glyoxysomes are a specialized peroxisomes. So if you take a cell, in a cell there is nucleus, mitochondria, Golgi complex, there endoplasmic reticulum, there are many organelles and there is also an organelle called as peroxisome. So these glyoxysomes are also a type of peroxisome, or you can call it as specialized peroxisomes, which contains a lipid bilayer, and it also occurs in mitochondria. So part of the cycle occurs in glyoxysome, part of it occurs in mitochondria, which is called as the powerhouse of the cell, and the rest of the reaction occurs in the cytoplasm or the cytosol. So cytoplasm or cytosol is nothing but the fluid matrix in which all the cell organelles is found. So this entire thing is called as cytoplasm. So it occurs in glyoxysomes, cytoplasm and mitochondria. It occurs in these. And one more feature of these glyoxysomes are they are always attached with mitochondria. They are found attached with mitochondria. And this glyoxylate pathway is also called as modified Krebs cycle, or it is also a type of gluconeogenesis. Why it is called as modified Krebs cycle, we will understand when we deal about the cyclic reactions occurring. And it is also called as type of gluconeogenesis. So that's about the introduction for glyoxylate pathway, where it mainly deals with the conversion of fats into carbohydrates. It is found in the plants, fungi, bacteria, lower invertebrates. It is completely absent in animals. and it is found only in the seedling stage. When the plant matures and two leaves are found, the photosynthesis occurs and this pathway slowly diminishes. It was discovered by Hans Krebs and Hans Korenberg in the year 1957. The species Pseudomonas species and using radioactive labeled carbon-14. It is found in three parts, that is glyoxysomes, mitochondria and cytoplasm. So if you come to the reactions that are involved in this, so what are the reactions that are involved in this glycoxylate pathway? So first we'll study about the glyoxysomes. So in glyoxysomes, what reaction occurs? So we all know that beta oxidation of fats. I said you fat gives rise to carbohydrates. So beta oxidation of fat gives a product, end product called as acetyl coenzyme A. This acetyl coenzyme A combines with oxaloacetic acid. Oxaloacetic acid. OAA means oxaloacetic acid. So acetyl coenzyme A combines with oxaloacetic acid in the presence of an enzyme citric synthetase. See, in enzyme nomenclature, we have to know that the first part it says about on what substrate it is acting. That particular enzyme on what substrate it is acting. For example, in this enzyme citric synthetase, this enzyme acts on citric or gives rise to citric acid. Synthetase means production, synthesize. So the enzyme itself says you, the name of the enzyme itself says you what function it performs. Citric synthetase means it is going to give rise to citric acid. It helps in synthesis of citric acid. Citric acid is also called as citrate. So that you have to bear in mind. So this enzyme citric synthesis, with the help of enzyme citric synthesis, acetyl coenzyme A, that is a product of beta oxidation of fats, combines with oxaloacetic acid to give rise to citric acid. The first compound is citric acid. Now this citric acid gets converted into its isomer form. That is isocitric acid. It gets converted into isomer form. What is this isomer form? Isomers are nothing but they have same molecular formula but they have the different structural formula. So if you see here, what is the difference between citric acid and isocitric acid? The difference is in the functional group. The functional group OH is attached in the second carbon atom of citric acid. But whereas in isocitric acid, the functional group OH is attached in the third carbon group. So it gets converted. Citric acid is converted to isocitric acid. To this reaction to occur, there is an intermediate form. So what happens is, first citric acid, it undergoes dehydration, means removal of water. It undergoes dehydration and in the presence of an enzyme aconitase, it forms aconitic acid. And this aconitic acid, again it takes more water. Hydration occurs to form isocitric acid. So the citric acid that is formed by the combination of acetyl coenzyme A and oxaloacetic acid is now getting converted to its isomeric form called as isocitric acid. Now what happens? This isocitric acid is cleaved with an enzyme isocitrate lyase. So the enzyme name itself, as I said, mentioned above, lyase. Lyase means lysis or to cut or to break. So what happens? This isocitrate lyase, it cleaves isocitric acid or it breaks isocitric acid into glyoxylic acid and succinic acid. So this isocitrate lyase cleaves or breaks isocitric acid into glyoxylic acid and succinic acid. What happens next? The succinic acid will come later. This glyoxylic acid, it combines with another molecule of acetyl coenzyme A and in the presence of malate synthetase. The name synthetase says it is going to synthesize. What is it going to synthesize? Malate, malic acid. So the name malate synthase. So this so formed glyoxylic acid, it combines with another molecule of acetyl coenzyme A in the presence of malate synthase to give rise to malic acid. This malic acid, now it undergoes dehydrogenation. D means removal. Hydrogenation means removal of hydrogen. So removal of hydrogen is also called as oxidation. So malic acid is oxidized or it undergoes oxidation in the presence of malate dehydrogenase to form oxaloacetic acid. So the oxaloacetic acid is rejuvenated or it is regenerated. This oxaloacetic acid again it fuses with another molecule of acetyl coenzyme A. It combines with another molecule of acetyl coenzyme A that is obtained from the beta oxidation of fats, and the cycle repeats. So acetyl coenzyme A, citric acid, isocitric acid, which gets cleaved into glycoxylic acid and succinic acid. This glyoxylic acid is getting converted to malic acid, and the malic acid is again getting converted to oxaloacetic acid. Again, this oxaloacetic acid combines with another molecule of acetyl coenzyme A, and this cycle repeats. So this process repeats. Now what happens to succinic acid that is formed? Succinic acid formed, it enters into mitochondria, that is the powerhouse of the cell. In mitochondria, Krebs cycle will be going on. So this succinic acid, it enters into the Krebs cycle and it forms oxaloacetic acid. How does it form? So first succinic acid is converted to fumaric acid. Then fumaric acid is converted to malic acid. Finally, the malic acid is converted to oxaloacetic acid. So if you just remember, succinic acid, it undergoes Krebs cycle to form oxaloacetic acid. That's enough. So succinic acid, it undergoes Krebs cycle in mitochondria to give rise to oxaloacetic acid via fumaric acid and malic acid. Now what happens? This oxaloacetic acid, it combines with ATP, adenosine triphosphate, which is the energy currency. So it combines with ATP. Oxaloacetic acid thus formed combines with ATP molecule to form phosphoenol pyruvate. So here in oxaloacetic acid there was no phosphate group. One phosphate group from here gets attached to oxaloacetic acid. This is adenosine triphosphate. Tri means 3, out of which one phosphate group is attached. So it becomes 2-phosphate. Hence the product will be adenosine diphosphate. And this oxaloacetic acid is a 4-carbon molecule. It is a 4-carbon molecule. It gets converted into phosphoenol pyruvate, which is a 3-carbon molecule. So there's one more carbon. That carbon comes out as carbon dioxide. So carbon dioxide is liberated. So when we do the seed germination, there's an experiment where respiration occurs, right? Carbon dioxide is liberated when the seed is germinating. So in there it is proved that carbon dioxide is liberated out. So this oxaloacetic acid combines with a molecule of ATP to form phosphoenol pyruvate, which is a 3-carbon compound, and carbon dioxide and adenosine diphosphate ADP molecules are liberated as byproducts. Now what happens? This phosphoenol pyruvate, which is also called as PEP or pyruvic acid. It is also called as PEP or pyruvic acid. Now it enters into the cytosol. Cytosol or cytoplasm both means the same. It is nothing but the fluid matrix in which all the cell organelles are present. So this phosphoenol pyruvate, it enters into cytosol and it undergoes a process called as gluconeogenesis. Gluconeogenesis is nothing but it is reverse type of glycolysis. So you should know what is glycolysis. Glycolysis is nothing but the production of glucose. So in reverse gluconeogenesis, what happens is, so glucose is produced from some other than carbohydrate is utilized. So the name, if you break down the name itself, you'll get gluco means glucose, neo means new, genesis means to give birth or origin. So glucose is given birth, or glucose is synthesized from a new substrate molecule other than the carbohydrate molecules. In glycolysis, carbohydrate is used. So in other than carbohydrate molecules such as PEP or phosphoenol pyruvate or pyruvic acid, such substrate molecules are utilized to form glucose or fructose. This process is called as reverse glycolysis or gluconeogenesis. So phosphoenol pyruvate, now it enters into the cytosol or the cytoplasm and a fluid matrix that undergoes gluconeogenesis or reverse glycolysis to form glucose or sucrose, which is the end product, that is nothing but your carbohydrate. Hence the aim is achieved where the fats are being converted to carbohydrate. Since it undergoes a gluconeogenesis, it is also called as type of gluconeogenesis, and since Krebs cycle is also involved here, it is also called as modified Krebs cycle. So these are the reactions that are occurring in this. Now, how to remember this entire reaction or entire pathway? So, for this, you have a mnemonic to remember. So the mnemonic is very simple. If you just remember that mnemonic, the entire cycle or the entire pathway will be in your hands. So the mnemonic that you have to remember is AO, AO6, AO6, and near G you write GAMO, not game or not momo, GAMO, and near S you write soap. So soap, GAMO, and AO6, like you have fig fruits and pig animal, like that you remember 6, AO6. So and GAMO and soap. If you remember these three, the entire pathway is very simple. So I'm going to show how to write the entire pathway from this mnemonic. AO6. A stands for acetyl coenzyme A. O stands for oxaloacetic acid. So oxaloacetic acid combines with acetyl coenzyme A, that is a product of beta oxidation. So it combines to form rise to citric acid. It combines to form citric acid. So C stands for citric acid and I stands for isocitric acid. Citric acid, then it is converted into isocitric acid. So it's easy, right? So the first letter indicates that respective compound of this. A stands for acetyl coenzyme A, O stands for oxaloacetic acid, C stands for citric acid, I stands for isocitric acid. Next, G and S. So isocitric acid is cleaved to form glyoxylic acid, and S for succinic acid and succinic acid. So AOCIGS, AO6, acetyl coenzyme A gives rise to oxaloacetic acid. Oxaloacetic acid gives rise to citric acid. Citric acid is converted to isocitric acid. Isocitric acid is cleaved to form glyoxylic acid and succinic acid. Now succinic acid enters Krebs cycle. It enters Krebs cycle to form O, that is oxaloacetic acid. So that the steps are not required in this pathway. So the succinic acid will undergoes Krebs cycle in the mitochondria to form oxaloacetic acid O, and oxaloacetic acid gives rise to P. P is nothing but phosphoenol pyruvate or pyruvic acid. So succinic acid SOP, soap. Succinic acid gives rise to oxaloacetic acid. Oxaloacetic acid gives rise to PEP, which is nothing but phosphoenol pyruvate or pyruvic acid, which is then converted into glucose or sucrose by the process of reverse glycolysis or gluconeogenesis. Soap is over. GAMO. Now this glyoxylic acid, it combines with another molecule of acetyl coenzyme A. Another molecule of acetyl coenzyme A. A stands for acetyl coenzyme A, to give rise to malic acid. It combines with glyoxylic acid, combines with another molecule of acetyl coenzyme A to give rise to malic acid, and this malic acid gives rise to O, that is nothing but oxaloacetic acid is regenerated. So this is what stands for GAMO, GAMO, GAMO. So if you remember, this is very simple. And soap, SOP, soap. Similarly, SIGS, AO6, SIGS, AO6. So it is just in a minute you can say. Acetyl coenzyme A gives rise to oxaloacetic acid. Oxaloacetic acid gives rise to citric acid. Citric acid is then converted to isocitric acid. Isocitric acid is cleaved into succinic acid and glyoxylic acid. Succinic acid enters mitochondria to undergo Krebs cycle, which as a result oxaloacetic acid is produced. The oxaloacetic acid gives rise to phosphoenol pyruvate. Phosphoenol pyruvate is then broken down by the process of reverse glycolysis to give rise to glucose or sucrose. And whereas this glyoxylic acid combines with another molecule of acetyl coenzyme A, that is a product of beta oxidation of fat, to give rise to malic acid. Malic acid gives rise to oxaloacetic acid, which again the cycle it helps in continuation of the cycle where it combines with acetyl coenzyme A. So this is our glyoxylic pathway. So remember this mnemonic, which will be very useful for you. AO6 and GAMO and SOAP. Now the final part or the conclusion part, what is the significance of this glyoxylate pathway? Significance. We have Many significance, out of which I have selected the most important significance. First significance is germination of seeds. It helps in germination of seeds. If this glyoxylate pathway was not present in seeds, there would be no energy for the seeds to germinate or sprout. So it helps in germination of seeds. And when we are discussing the entire pathway, we came into account of two enzymes, isocitrate lyase isocitrate lyase and malate synthetase. We came into these two enzymes. So these two enzymes are important because they cause pathogenesis. They help in pathogenicity or pathogenesis. So if this isocitrate lyase enzyme and malate synthetase enzyme are present in fungi or bacteria, they cause disease. They help in pathogenicity. So if the concentration of this enzyme is more, the pathogenicity is also more. So this was the thing. So an isocitrate lyase enzyme concentration is more and malate synthesis enzyme concentration is more. They help in pathogenicity. So several experiments was conducted regarding this era. One of this was where Sharma et al. In Mycobacterium tuberculosis. This Mycobacterium tuberculosis causes the disease tuberculosis in mice. So what they did was they attacked this enzyme. And they inactivated it. So it is like switch on switch off mechanism where they inactivated this enzyme. When they inactivated this enzyme, the disease or pathogenicity reduced in mice. So Sharma and et al. They conducted several experiments where one of the experiment was they found out an anti-tuberculosis drug or the antimicrobacterial effect where this drug was deactivated or inactivated. The drug was given to inactivate this enzyme. So isocitrate lyase and malate synthesis enzyme was deactivated. As a result, the tuberculosis in mice decreased. This was one of the experiment. And one more experiment was Mycobacterium grigia. Mycobacterium grigia causes rice blast disease. So what they did was they switched off this ICL gene itself. ICL gene is present in this Mycobacterium grigia. ICL stands for isocitrate lyase gene. So this ICL gene, if it is present in Mycobacterium grigia, if it is activated or if it is expressed, it causes rice blast disease. If they switch off, if this gene is deactivated and it does not cause rice blast disease, this was also further proven in Candida albicans, which is a fungi. So in Candida albicans also it was further proved experimentally when they switch off ICL gene, the disease or the pathogenicity is reduced much. So this was also one more experiment. And at last in Salmonella species also they conducted the same experiment. So Salmonella is a bacteria that causes disease and mice it causes chronic infection. So when they turned off this ICL gene, what happens is this chronic infection was reduced. So these are the significance. This can be considered as clinical significance, but very important is they help in germination of seeds. And as I said the recent studies have shown that this enzyme are present in hibernating bears. Bear animals. We know that bear is a thick-skinned animal. It undergoes hibernation during the winter period. So what it has to produce energy, right? So what happens when it undergoes hibernation? It has very thick skin and it has a huge amount of fat reservoirs. So only during hibernation or it occurs is this cycle occurs, the Glyoxylate pathway cycle occurs where it produces energy. And when the hibernating stops, this cycle also stops. So this is the most recent study where they came to conclusion that in hibernating bears also the glyoxylate pathway occurs. And what happens? This pathway is absent in humans, as I said. But this glyoxylate crystal, we should know this. Going a step forward, this is something to do with glyoxylate crystals. If the glyoxylate crystals are present in human bodies, it gets converted into kidney stones. It is salt. It does not easily gets dissolved in water. So only if they say you have to drink more plenty of water. So what happens in the less amount of water, this glycolate aggregates and forms clusters or stones called as kidney stones, and it gets accumulated in your kidneys and is not removed by the urinary blood. It's not filtered because it lack of water. So this glyoxylate crystals aggregates to form kidney stones. That is one of the important thing. And the other thing you have to remember over here is this glyoxylate. If glyoxylate increases, sugar content also increases in blood plasma. Sugar content also increases in blood plasma of human being. And if sugar content increases, we all know it leads to a called as hyperglycemia. It leads to hyperglycemia or type 2 diabetes. Type 2 diabetes. So this is what happens when glyoxylate increases. Sugar content also increases the blood plasma and causes hyperglycemia, type 2 diabetes. So this glyoxylate pathway plays a very important role in plants and other microorganisms involved in pathogenicity. So this is about glycoxylate pathway. I hope this video was very useful for you. So please comment, please share this video, please like this video. Thank you.